Chapter 1. Genetic Basis of Inheritance. Dear students, we are all living organisms. We get, that means we inherit, some characters, and these characters are called as traits, and these traits we get from our parents. And in turn, we transfer these traits to our offsprings, that means to our children. And therefore, offsprings resemble their parents and they resemble each other also. But if you observe minutely, you will definitely notice the differences also. So what is meant by heredity? It is defined as transmission of characters from one generation to the next or from parents to offsprings. Variations are the differences between the parents and offsprings, among the offsprings of the same parents and among the individuals of the same species. Genetics is the branch of biology that deals with heredity and variations. Now the first scientific explanation regarding inheritance was given by Mendel, and the principles are now popularly known as Mendel's laws of inheritance. So let's know about this. The organisms produced by asexual reproduction or plants produced by vegetative propagation are identical to their parents and can be called clones. Organisms produced by sexual reproduction are called offsprings, and they are not identical to either of their parents but inherit some of the characteristics from both the parents. The characters, those are transmitted from one generation to the next through particles, are called factors by Mendel, and now these are known as genes. Now we'll see about Mendel's experiment. Mendel selected garden pea, that is Pisum sativum L, for his experiments because of the following characteristics. Garden pea is an annual plant and completes the life cycle within 3 to 4 months. Due to the short lifespan, he could grow 3 generations in a year. It is a small herbaceous plant that produces many seeds, so he could grow thousands of pea plants in a small plot. It is naturally self-pollinating and was available in the form of contrasting characters. For example, tall and dwarf plants, yellow and green seeds, violet and white flowers. There were no intermediate characters. Now genetic terminology. Character, it is a feature of organisms. For example, height of stem. Then trait, an inherited character and its detectable variant, that is tall or dwarf. Factor, a particle present in the organism which is responsible for the inheritance and expression of a character. Then gene, it is a particular segment of DNA which is responsible for the inheritance and expression of that character. Then alleles or allelomorphs, the 2 or more alternative forms of a gene are called alleles of each other. Then dominant, it is an allele that expresses its trait even in the presence of an alternative allele. Then recessive, it is an allele which is not expressed in the presence of an alternative allele. Then phenotype, the external appearance of an individual for any trait. For example, in pea, for height of the stem, tall and dwarf are the 2 phenotypes. Tall is determined by capital T or capital T small t. The genotype, it is representation of the genetic constitution of an individual with respect to a single character. For example, in pea, tall plants can have genotype capital TT or capital T small t, and dwarf will have small tt. Then homozygous, an individual possessing similar alleles for a particular trait. These are homozygous or pure for that trait. Then heterozygous, an individual possessing dissimilar alleles for a particular trait are heterozygous or hybrid. Then pure line, an individual which is homozygous or true breeding for one or more traits. Then hybrid, it is heterozygous having one or more contrasting traits. Monohybrid, it is heterozygous for one trait. For example, hybrid tall produced in a cross between pure tall and dwarf parent. Then F1 generation, the progeny, that is offsprings produced from a cross, is called first filial or capital F1 generation. F2 generation, the second generation, that is progeny produced by selfing, that is interbreeding of F1 generation offsprings. Then Punnett square or checkerboard, it is a diagram that is used to show possibilities of combinations in a particular cross or breeding experiment. Then homologous chromosomes, the morphologically and structurally similar chromosomes present in a diploid cell. Now this is the classical method of symbolization. Parental generation P or P1, first filial generation F1, second filial generation F2, third filial generation F3, dominant capital T for tall, capital Y for yellow, recessive small t for dwarf, small y for green, female this symbol, male this symbol.